Okay, then we can insert it, okay?
Okay. I'm going to try my best to stay on time so we can all get lunch. Just make sure this works. Great. So a little background, because I'm changing topics slightly from what we've been discussing. My lab has become mainly interested in pediatric cardiology or congenital heart disease. So this is the most common type of birth defect, leading cause of death in infants. This affects about 8 to 9 out of every 1,000 live births. It can be very mild, requiring just some medication, or it can be very severe, requiring 3 or 4 or maybe even 5 to 10 complex surgeries. So due to increased survival, there's about 1 million people now living with congenital heart disease. It used to be a death sentence. Now most of these children make it into adulthood. And so there's other forms of cardiovascular disease for children other than congenital, which things like myocarditis, all these acquired dilated cardiomyopathy, hypertrophic cardiomyopathy, but basically the outcome for cardiomyopathy for children is essentially the same as it was 30 years ago once they survive it. So one thing that my lab is interested in is hypoplastic left heart syndrome. This is one of the most common single ventricle defects. You can see it's about 2 to 10% of all births. It's underdevelopment of the left heart, and you can see here's the normal LV and normal heart of a patient here. In your hypoplastic left heart syndrome, there's issues with the left ventricle. Several of the valves are defective. This was basically 100% fatal about 30 years ago, and now they have surgeries that can essentially turn the right ventricle into the systemic ventricle, which is great because these children now survive. About 60% of them survive the first surgery or first 2 surgeries, but this increased load on the RV, it's meant to supply blood to the lungs. It now has to supply blood to the entire body. It creates an increased pressure and volume overload, and these children eventually go into heart failure. So you can see here in cases of RV dysfunction, 18-month survival is about 35%, and that's shown here. You can see once you have the grade of RV dysfunction gets more severe, the survival goes way down. This is transplant-free survival. These children also have a variety of other issues here. Here's exercise capacity, and you can see Fontan repair, which is the 3rd surgery. These children have essentially half the exercise capacity of their counterparts. And then if you look at IQ, if you look at the 100 being sort of the median IQ of children, most of these children cluster below the average IQ due to the repeated surgeries. So there's regenerative therapy is greatly needed. One thing that my lab studies, which is cardiac stem cells or cardiac progenitor cells. These cells were discovered in 2003, tissue resident. I'm not going to get too much into them here. We isolate them from C-kit beads from atrial appendage biopsies from children. And so you can see they differentiate maybe into some lineages. I'm not going to get into that debate here. But for the context of this talk, our cells are C-kit positive, NKX, and GATA4 positive. I'm really cellular agnostic, but once again, that's not here or there. So we had a paper maybe 2 years ago showing that if we took these pediatric cells from these biopsies, they actually had differential effects based on their age. And so you can see here, we gave these animals RV dysfunction by banding the pulmonary artery, and you can see TAPSE, which is a measure of RV dysfunction. These animals are low. If you give them cells from a child, which we deem to be greater than a year old, you can see there's really no improvement here. If you gave them cells from an infant, in blue, you can see there was no improvement initially, and then these animals started to improve. But if you gave cells from a neonate, which we said was within the first week or 2 of life, you can see now there's a significant improvement in RV function early, and it even went up over time. And this is shown here in RVEF on the y-axis with time after surgery here. And then also we looked at wall thickness, and these neonate cells, these cells derived from neonates, improved wall thickness. And so this is great and all, but what my lab is really interested in is the numbers game. Right? Because as Roberto showed, that you need lots of different cells, you need to have repeated injection, but we think that the cells also matter. And you know, my cells might be very different from your cells, which might be different from everybody's cells. And so we became interested in actually using regression analysis and using what we call partial least squares regression. What we can do here is we can identify microRNAs that are in cells, genes that are in cells, or exosomes, the different conditions that these were derived in, and then we see what effects they have. And ultimately, you come up with a regression coefficient matrix where you can then get new data. You can plug in your new data and see what you would expect these responses to be based on your loadings. And so in this paper here, you can see we identified clusters of genes which we thought were important or regulating this response. Some of them were very important. You can see there's CXCL2. A lot of inflammatory genes came up, showing that these neonates have higher levels of inflammatory genes that seems to correlate with improvement. Once again, this does not show causality. It's really just a hypothesis-generating tool. And then things like collagen, which we showed that collagen was downregulated in these animals. Collagen is actually downregulated in these cells as well. So a lot of what we're seeing in the model actually holds true in the cellular response. But one thing that we're really interested in is that we can create predictive models. And so I mentioned this is a regression fit. And so what you can do is you can say, well, we want to see what TAPSE is or fibrosis. We can actually come up with a regression fit and fit data to our model. And so you can see here we have a high R squared, 96%, 93%. So we can train our model on the signals and then use the regression analysis to fit data to the model. So our original model was 9 patients. And so now in this expanded one, we've done 40 patients. And you can see here neonates in purple, infants green, red child. And you can see here there are some that fit the model and some that don't. And so what we can do then is we can find out here, there's a patient here, 1065, who's an infant, who's a child who doesn't seem to fit with these other children. There's also a neonate in purple here, patient 1050, that doesn't fit with his neonate counterparts. And this is looking at predictive functional improvement. So if we plug the model in, we can see that the predicted functional improvement of the neonates doesn't fit with the other ones. And so we actually can go back and we can say here in red, here's what we predict. For instance, this is proliferation. We say, what do we expect the proliferation of these various cell lines to be? And then do our data fit the model? And in fact, it looks like for the most part, our data fits the model. So that's pretty exciting. If we look at principal component analysis, you can see here that the CDCs and CPCs from another trial, this is from Sanjay Kaushal's data, we can actually plug it in with our neonates and infants and childs, and we see that they're different, which is good, right? We expect adults and children to be different. The bad news is that they're very different from our cells, so we really can't compare them. But what we can do then is we can look at exosomes, and that's essentially what we did in our study, is we looked at exosomes from these models. So we created an additional model based on our Circ research paper in 2017, where we had exosome microRNA. And you can see here, we can predict ejection fraction, fibrosis, and angiogenesis with fairly high predictability. So if you look here, the predictability here is on the y-axis, and here's our various outputs, EF, fibrosis, and angiogenesis. Interestingly, in this study, what we did with Dr. Kaushal is we isolated exosomes. These are cells. These are cells derived, either exosomes derived in vitro or from the serum. So he injected the animals with the cells and then isolated exosomes from the serum. And you can see here, whether we use exosomes from in vivo or exosomes from the serum, it doesn't really change our predictability. We can still predict with very high fidelity what's going to happen. So the interesting thing is that he went and he did this study here. So this was independent from our laboratory. We're just a data aggregator. We collected all of his data. And so he compared CPCs to CDCs in this study. Remember I said I'm data agnostic or cellular agnostic. And you can see here, if we look at the predicted responses in red here, if we use the exosomes from the serum and we compare them to the exosomes from the in vitro culture, you can see here the serum exosomes actually predict what's going to happen with much higher fidelity than if you collect the exosomes in vitro. And so this is actually not very surprising if you believe that the microenvironmental controls what the cells release. And so you can see here, here's EF, here's fibrosis. This also holds true for angiogenesis. And you can see there's actually a pretty big gap. So if you were to isolate exosomes from the media and sequence them and try to make a prediction, you would essentially be, staring into the wind trying to figure out what's going on. But here, if you can isolate serum exosomes, if you can treat a patient with cells and then isolate exosomes from the serum, that actually does a better job of telling you what's going on. And so this is shown here, principal component analysis. You can see here, if we compare the in vitro exosomes derived from CDCs and CPCs, they look almost identical. We could find very little different about them. However, when you now look at these derived from the serum, you can see they now start to look a lot different and actually more predictive. And so they're very different. And this confirms our prior study in our Circ research paper showing that if we made these cells hypoxic, they actually drastically change what they put in their exosomes. And so this is exciting to us because this is data from somebody else's lab that we're then able to synthesize, make predictions, and predict what's going to happen in his lab. And then we can go look at these exosomes and we can say, using our regression analysis, we rank the microRNAs that covary together, and we say how likely is a given microRNA to contribute to a response. And so you can see here, if you map out the responses, here's angiogenesis, here's ejection fraction fibrosis. Now you can see microRNAs that rank near angiogenesis, that rank near fibrosis, and now you can go back and you can test this in vitro, and you can say, does MIR 1256 or MIR 525 induce angiogenesis? So you can generate new hypotheses based on these predictive models. And so in conclusion, exosomes pack a similar punch to the cells and are actually more predictive than examining the cells themselves. So I didn't show this, but when we used his cellular microRNA and cellular RNA-seq data, it was actually a horrible predictor of what was going to happen. We could predict with almost no fidelity what was going to happen in his study. And so we can use our model to predict functions from other studies based on RNA-seq and array data. If we can't match the outcomes, that means if you're studying something different than we're studying, we can't do it as well, but we can still try. Cells produce different exosomes when delivered in vivo, and these are more predictive. And then we can create aggregate models to include other cells, and it's what we call quantitative predictive medicine. And so you can see here, ultimately, we want to push this paradigm they call the Q signal response paradigm, where you have different cues, which could be different cells, different variables contributing to the cells. So we're interested in age and sex. You collect the data, you look at the signals from the data, you have your in vivo, in vitro, and clinical data, and then you can actually map out how each response varies the signal, which leads to this ultimate response here. And so where are we getting at next? Well, this is a material a part of it as well. And so what my lab is also interested in is printing exosomes. And so here's a recent paper we're about to submit here from one of my students, where we're taking cells and exosomes and using gelatin methacrylate and some other materials here, we're actually able to print really nice strands of gelatin methacrylate patches. This contains other things like that, containing exosomes. And you can see here's what these patches look like. And you can see we get pretty good distribution of exosomes across the patches. So we're pretty excited about being able to tailor what's going into the patches and then predicting what's going to happen based on the inputs. And so with that, I'll thank the people in my lab who did this. We have a lot of collaborators, including Sanjay Kaushal at Maryland, Mark Sussman and Josh Hare, who have given us a ton of data to try to predict what's going to happen in our clinical trials. And it should be noted that we actually have a clinical trial we're about to start enrolling patients in, delivering CPCs to hypoplasts. This IND has been granted by the FDA between Josh Hare, Miami, and our institute. And we're hoping that we'll be able to predict what's going to happen to these patients before we give them the cells, and then use that data to see what happens next. Thank you. 12 minutes.
Thank you, Michael. Is there a quick question and time for a quick answer?